Hey everybody, welcome to your Chapter 3 Infection Control lecture. So we are going to be talking about, let me get back to the objectives, quite a few things here. So identifying the chain, the 6 links in the chain of infection, different ways in which diseases can be transmitted, that we want to make sure that we learn proper hand hygiene, how we prevent the spread of infection, including personal protective equipment, and ways that we can make sure we keep our patients safe and ourselves and our coworkers as well. So some things to know, infection control is the process of reducing exposure to pathogens and spreading disease. All healthcare providers are responsible and must take the steps necessary to prevent the spread of infection. Microorganisms, of course, are germs, the fancy word for germs, tiny little things you can only see with a microscope. We have germs all over our bodies. Not all of them are going to cause disease. Many of them we actually need, especially think about your gut. We have a lot of microorganisms inside our gut. We need those to help keep us healthy. So they're not all bad, but sometimes we get germs, microorganisms, or pathogens that can give us a disease, and then also we can spread it, of course. Microorganisms or germs need a few things to be able to survive. They need food, moisture, darkness, a suitable temperature, and then some germs need oxygen and others do not. If they need oxygen to live, they're called aerobic, and if they don't need the oxygen, they're called anaerobic. And we'll talk a little bit more about this when we get into the chain of infection in a minute. Here we go. So the chain of infection, well, first of all, and we're going to talk more about hand hygiene in a minute here, but most infections can be prevented using hand hygiene. And something that's really important to keep in mind is if you, as a phlebotomist or as any healthcare provider in any way, can break one of these links in the chain, you can prevent an infection from spreading and occurring. So let's break it down now. Let's talk about these different links in the chain of infection. Let me skip ahead. Bear with me. I want to see. Okay. So we've got the first link. That's the infectious agent. The infectious agent is that germ, that pathogen, or a disease-producing microorganism. Number 2, the second link is the reservoir. Reservoir is a fancy word for a germ's home. This is where the germ lives, grows, and multiplies. So this can be us, this can be animals, water, food, or air. All of those things can be a reservoir. Number 3, the third link, is the portal of exit. This is how or where the germ leaves the reservoir, leaves its home. So when we're talking about our bodies, this can be the skin, respiratory tract, GI tract, any of our mucous membranes like our eyes, our mouth, etc. The mode of transmission is the fourth link in the chain. This is how pathogens travel. So different germs and pathogens are going to travel in different ways. It can be contact, droplet, or air, and we're going to talk about that in a little bit. And you can also get these germs transmitted to you or transmitted to other people via indirect or direct contact. And we'll also talk about that a little bit more when we get down to transmission. Number 5, the fifth link in the chain of infection, is the portal of entry. Easy to remember, it's the same as the portal of exit. So germs get in the same way they get out. So again, skin, respiratory tract, our GI tract, etc. Finally, the last link in the chain of infection is number 6, the susceptible host. This is a person who is at risk for developing an infection from that germ. Some things that put us at more higher risk for being a susceptible host is being immunocompromised, having an open wound, a drain or a tube, because it's easier to get in, another place for germs to get in. If we have poor nutrition, so our body is weak, it doesn't have the ability to fight off these germs. Chronic diseases, those also just weaken our immune system, our body over time, make us more susceptible, and the very young and the very old. We do have some natural body defenses, and as we get into I think it's chapter 5, we'll talk about some of these natural body defenses, especially in relation to the skin. But the skin is our largest organ. It covers all of our body. If it's intact, it's protecting us. Mucous membranes, these are the lining of any body opening, right? Our mouth, our nose, eyes, etc. We also, these are here to protect us. Lymphocytes, we're going to talk more about lymphocytes and antibodies as we get into the chapter on, forget which, yeah, chapter 5 again. We talk about the different body systems, but basically our body is designed to help fight off invaders, foreign invaders, pathogens. Lymphocytes help protect our body, antibodies protect our body. That's part of why we get immunized, to build up antibodies so that when we do get introduced to a foreign substance, our body recognizes and says, I've seen you before, I know how to fight you. Immunity in general just means our resistance, our ability to fight disease. Some signs of infection, redness, heat, swelling, pain. I'm sure you guys have all had a little spot maybe on your body or something where you got a cut and it's become infected. You might not have it to the extent of this picture, but you've seen that redness, that heat. When you just touch it, it feels a little bit warm, sometimes the swelling and the discomfort. You might even have things like pus formation, a little bit of wound drainage as well. Let me hop ahead here. So standard precautions. These were things, or I should say the steps, the precautions that were introduced in the late 1980s to help protect healthcare workers from exposure to bloodborne pathogens. We talked about bloodborne pathogens already in the previous week, and we talked a little bit about standard precautions there. So just a little reminder, I think I mentioned in previous lectures that standard precautions in the 80s first came about because of the HIV and AIDS outbreak. It was developed by the CDC. It was a guideline to apply to all body and blood and body fluids. And it applies to all healthcare providers, not just phlebotomists, not just nurses, et cetera. It's all healthcare employees who may be exposed to any body fluids, secretions, and excretions, with the exception of sweat. The CDC advises that healthcare employees also don't wear artificial nails. I think I mentioned that a little bit too, because those are a great place to harbor germs. Long nails, artificial nails, or polished nails, even if it's clear polish, great place to harbor germs. In 1996, the CDC took these standard precautions a step further to include isolation precautions. And I think, did I pass that already? We're going to come up to the transmission precautions in just a few slides here, okay? So we're going to back up a little bit to hand hygiene. We said earlier how this was the most important step that we can take in stopping the spread of infection. Hand hygiene, let's see, is important in preventing not only infections and germs out in our, when we're out in the community, out in our homes, but they're also important in preventing healthcare associated infections. Healthcare associated infections, or you'll see abbreviated HAIs, are infections that occur while a patient is hospitalized or is receiving treatment for another condition in any type of healthcare facility. So this could be in the hospital, in an ambulatory care, a nursing home, etc. Remember, protecting the safety of our patients, our colleagues, yourself, and even your families is a huge aspect of your career as a phlebotomist. Hand hygiene includes both hand washing with soap and water and using the alcohol-based hand rubs. You do need to make sure that you know when it's appropriate to use alcohol-based hand rubs and when you must use soap and water. And I'll talk about that a little bit when we talk about some of the transmission-based precautions, but also make sure to review that in your text to know when it's appropriate, okay? Soap and water, a few of those examples. When you want to use soap and water for sure is when after you've used the bathroom, when you've worked with patients with a germ called C. diff, if your hands are visibly soiled. And they also say after you've used hand sanitizer 3 times, really that 4th time should be soap and water. There are steps to hand washing. I know that you've done this in your in-person classes with Michelle, but these include the steps that you see here. I'm not going to review all those steps right now because I know this is something you're doing in person. So make sure that you review this. One of the most important things to keep in mind or to take away from the steps in the hand washing is wash for at least 20 seconds and make sure that you're getting all surfaces. The alcohol-based hand gels, one of the things to remember about this is it should take you between 15 and 30 seconds for that hand sanitizer to fully dry. If you are feeling that it's drying before the 15 to 30 seconds, you're not using enough. You have to give it enough time, just like we have to give enough time to hand washing with soap and water. And we just talked about that, about keeping the nails short and clean, no artificial nails. Another thing that we need to make sure that we do to include infection prevention is our jewelry. We need to be aware of that, bands on our fingers. We don't want to wear more than one single solid band. Anything that has protrusions, anything like, different stones or diamonds within the band, or if it's intricately kind of carved, that's a great place as well for germs to live and grow. And just natural cracks in our nails. That's why it actually is important that we actually have not only hand hygiene, but we take good care of our hands, keep them from getting dried out too much, which can be quite a problem when we're washing our hands as frequently as we do as healthcare workers, but it's something that we've got to pay attention to. Another thing to remember in the spread or preventing the spread of infection is covering your cough. You want to cough into the upper arm, into your elbow. That's to protect you from spreading it into your hands, which you then could touch something, a surface or a person. And of course, always washing our hands if we do accidentally cough or sneeze into them. So let's bounce back to those isolation precautions I talked about. We already talked about the standard precautions. Now we're going to talk about isolation precautions in different modes of transmission. So contact is one way that germs can spread. There's also contact precautions. So let's talk a little bit about that. Contact is the most frequent source of healthcare-associated infections, or the HAIs. This occurs by either direct or indirect. I said I would mention that again. Direct is the physical transfer of pathogens from a reservoir, so the germ's home, to a susceptible host. So that could be as simple as me touching you and spreading the germs. Indirect is when a non-living item is handled prior to contact with susceptible host. So this can happen in the laboratory setting, in the outpatient lab. This can happen in a nursing home if you're working around there, drawing blood. You touch a contaminated piece of equipment, you don't wash your hands, you go into the next room, and then you touch your next patient, and you have just cross-contaminated. That would be an example of indirect. You're touching, again, a piece of equipment, maybe a soiled linen, and then you touch that next susceptible host. Let me see. Bear with me. Okay, so when you believe that you may come into contact with anything contaminated beyond the gloved hand, because remember, standard precautions includes wearing gloves. If you think you're going to be coming to anything that can be spread by contact, an example of this is the C. diff. MRSA is also spread by contact. You're going to have to take the additional protection and PPE of adding a gown on top of your gloves. Okay. So droplet, that's another way that germs can be spread, and it's another type of isolation precaution. Droplets spread when a droplet from an infected person is propelled at a short distance. So germs that are spread by a droplet typically don't travel any further than 3 feet, typically. They enter the host typically through the mucous membranes, spread by coughing, sneezing, breathing, or talking. This can also occur with specimen handling as a phlebotomist. When you have droplet precautions, you have your gloves, your gown, but you've also added goggles or face shield or mask. An example of something that is spread by droplet would be our yearly influenza or flu. Okay, so airborne. Airborne is when small particles can carry the pathogens for further distances. They can travel by air, in currents, in dust, so they spread much further. We breathe these airborne pathogens in. It can also happen when aerosols are created. So certain procedures, certain processes in the processing of lab specimens, like the removal of a tube, can produce aerosols. This is when you might be working under the fume hood in the lab, but aerosols or anything that can carry and spread pathogens in the air can spread these things. So when we move from wearing a surgical mask to like an N95 or a respirator type mask. Tuberculosis and measles are an example of something that is airborne and can be spread through the air. Let's see if I'm missing anything from that. I don't think so. Okay. So you need to know or be ready to recognize these signs. So in most facilities, if someone or if you have a patient who is on any one of these precautions, whether it be contact, droplet, or airborne, you're going to see this sign posted typically on the outside of their door or either on the door itself or right on the side of the door. This is your warning sign. Hey, stop, see what's going on. What is the PPE that you need before you walk into that room? And let's talk a little bit more in detail about PPE. And let me find my notes so I don't forget anything here. So PPE is a mandate by OSHA, and they mandated this, of course, to help minimize the exposure to bloodborne pathogens and other airborne droplet, contact pathogens as well. So PPE is includes gown, gloves, mask, protective eyewear. And when we say mask, of course, we need the surgical or the respirator if necessary. Whenever blood or body fluid from one person comes in contact with another person, there's of course the major risk of bloodborne pathogen exposure. When we're talking specifically about bloodborne pathogens, we're most concerned with hepatitis B, hepatitis C, and HIV. So hepatitis B is your most worrisome one. It's the one that's most easily spread. Hepatitis B can actually live on surfaces for up to 1 week and still infect you. So keep that in mind. In general, when you're using PPE, you don the PPE, and don means just to put it on. You don PPE before entering the room and at least before you have contact with the patient. You need to make sure that you apply your PPE in the proper sequence. And I know that this is something that you've reviewed in class as well, but I'm going to go ahead and review it one more time because it is so important. The correct sequence to apply your PPE is going to be gown, a mask or respirator, a goggles or face shield, and then your gloves. You want to use your PPE carefully so that you avoid contaminating yourself, others, or spreading the germs. And then you want to remove the PPE either right at the doorway before you step out or just immediately outside the room. The exception to that is a respirator. You always remove a respirator just outside the room. Because if you remove that respirator and you're still inside the room, you're at risk of catching that germ. And just like when we're applying PPE, you want to make sure you remove PPE or doff PPE in the correct sequence. You want to start with the most contaminated. You want to remove gloves first, then your face shield or goggles, then your gown, and last your mask or respirator. Anytime you remove PPE, you immediately want to perform hand hygiene. And there it is, just in writing in the order of which you would apply. And you'll have plenty of practice with this as well. I know that says references. I have a couple more things I'm going to talk to you about before we move on. I also want to talk about some drug-resistant bacteria because these are things, and I mentioned like MRSA and the C. diff, those are all drug-resistant bacteria. Several bacteria have become resistant to antibiotics, unfortunately, and this has a lot of factors, but some of the reasons include, overuse of antibiotics or not completing your antibiotics when they are prescribed to you. Extra precaution is taken when patients who are infected with these certain drug-resistant pathogens. Like I mentioned, MRSA or methicillin-resistant Staphylococcus aureus is one of these. This is difficult to treat. It can lead to tissue loss and sepsis. Sepsis, of course, is a life-threatening response of the body to infection. It's an infection throughout the body. MRSA can be transmitted from person to person or object to person. You've maybe heard of vancomycin-resistant enterococci. This is VRE. That's another example. The Clostridium difficile or C. diff. This is a spore-forming bacteria that causes diarrhea. It can live outside of the human body for a really long time, so of course means it's easier to spread. Can be transmitted from person to person or object to person. MRSA and C. diff are ones that you will see spread very quickly, especially in settings such as the nursing home, because we have a lot of susceptible hosts living in close quarters, sharing staff and equipment. C. diff, for example, especially in the older population, can be life-threatening because of the amount of diarrhea, dehydration that these individuals can deal with when they get C. diff. Let me see what else here. There's one other type of precaution that I didn't talk about earlier when we talked about droplet, airborne, and contact. There's actually one other one that you may not have heard of. It's called protective environment, or PE. It used to be called neutropenic precautions. What that is precautions that are used for patients who are severely immunocompromised, and they're super susceptible to getting germs from us, even if we're not visibly ill. So think of your cancer patients. That's a really good example for this. They're so immunocompromised that there's not necessarily anything that we're going to catch from them, but they're so susceptible to catching things from the outside environment or from us that we actually gown up, glove up, mask up before going into their rooms to provide extra safety for them. So this is an important thing that we need to remember as phlebotomists. If we see that somebody is on these protective environment precautions, we need to make sure that we're doing our role in not bringing anything into those patients. You'll also notice in those rooms, they can't have things such as, a lot of people like to deliver flowers to people in the hospital. They can't have flowers or plants, certain foods they can't have just because of the increased risk of pathogens from those things. A couple things to keep in mind as phlebotomists. You never want to take a tray of phlebotomy equipment into an isolation room. You want to take only what you need for that particular draw. If you forget something and you've already donned your PPE, you've got to doff it, go through that whole process again. You can't go into a room with PPE, walk out because you forgot a supply and walk back in. Yes, that would be much quicker, but you can't do it. You got to take the PPE off, go out, get your supply, put new clean PPE on. Any unused equipment that you take into those rooms, you're leaving in there. So again, you want to come fully prepared, but you don't necessarily want to bring excess equipment either, because that equipment is going to need to stay in the room. There is a procedure called double bagging that you will use as a phlebotomist when you're drawing labs in rooms of patients with contact, droplet, or airborne precautions. And what that is a way to prevent, we had to get those specimens out, right? So you've got to get those specimens out, but you don't want to contaminate. So the way it works is 2 biohazard bags are used. You're the phlebotomist in the room, you're wearing your full PPE, you place any items for disposal or any specimens that are going to leave the room into, to be 2 separate bags. So let's just use one. Let's say we're having to dispose of items outside the room. We'll use that example. So you have some items that you need to dispose of outside of the room. You're in the room in your PPE, you put the items for disposal into 1 biohazard bag and seal it. You have a second person waiting just outside the door with a second biohazard bag. So when you're ready, they're going to be standing there with that biohazard bag open. You're going to carefully place the first biohazard bag within that second one without contaminating the outside of that second bag. Then it will be sealed by the person outside holding it, and will be transported or brought to the proper place for disposal. Again, that's done in contact, droplet, airborne isolation rooms of any kind, so that we're not bringing germs from inside the room outside the room. So another main point to take away from this, we follow standard precautions with every single patient. We assume that every single blood or body fluid could potentially be contaminated. So that's why standard precautions are in place. All right, and then transmission-based precautions as necessary. All right, I hope you guys had a good understanding of all of that. If you have any questions, let me know, and I'm here to help you. And I think that's it.